Hello everyone out there. So let us start with today's important topic of oogenesis. So we have completed the gametogenesis part. In that we have completed the spermatogenesis and spermiogenesis. So now we are going to start with oogenesis. So without further delay, let's get started, and please don't skip the video in between because many important topics I'll be discussing in the middle and at the end of the video. So please go through the full episode. And if you have not yet gone through the previous episode, I am putting the link down in the description box as well as in the I link above. So let's get started. So let us come to the proper definition of oogenesis first. Let us see that oogenesis, what is it? It is the process of formation of female gametes. The process begins inside the fetus before birth. We have told already that it is formed inside the fetal ovary itself. The steps in oogenesis up to the production of primary oocyte occur before birth, and the primary oocytes, they do not divide further. They stay in that arrested state, that is, they are arrested in the prophase 1 of meiotic division. So prophase 1 of meiotic division, they are arrested in that state, right? So they are arrested in the prophase 1 of meiotic division. They either become secondary oocyte or they degenerate. So during the process of we can say birth till puberty, there are several primary follicles which degenerate, but few are still there which become secondary oocytes after puberty. Okay, is it clear to everyone? Now, oogenesis occurs in the outermost layer of the ovaries. So what is the place of occurrence of oogenesis? It is occurring in the outermost layers of the ovaries, and oogenesis starts with a germ cell called the oogonium. Okay, and undergoes the mitosis to increase in number. So when it is undergoing mitosis, it will increase in number and divide into more number of oogonium. The process of oogenesis takes place in 3 different steps. First is prenatal, then is antral, and next is pre-ovulatory. So prenatal means in the fetus state. Okay, so it is prenatal. Then comes the antral part, when the antrum or the fluid is formed. And then comes the pre-ovulatory, that is before the ovulation the stage is coming. So we will see all the stages and details, and then we will move on to the diagram. So 3 stages we have discussed in this oogenesis process. The first stage is the prenatal stage. So what is happening in this stage? The primary oocyte, which grows while being arrested in meiosis 1, it starts growing in the prenatal state. Okay, the follicular cells, they proliferate and they form a stratified cuboidal epithelium. So the follicular cells, they start forming a stratified cuboidal epithelium, and such cells are known as granulosa cells. So this we will see in the diagram of the ovary in the next slide itself. So these cells, they secrete glycoproteins to form zona pellucida around the primary oocyte. So the stratified cuboidal cells are known as granulosa cells, and they secrete glycoproteins which form the zona pellucida around the primary oocyte, right? So let us move on to that part. So what is happening in the antral stage? We have told in the previous state, that is in the pre-antral state, that the zona pellucida is formed. Now in the antral state, the fluid filled area present in between the granulosa cells which combine to form a central fluid filled space, that is called the antrum. So what is antrum? It is the central fluid filled space. Okay, now they are known as secondary follicles. So secondary follicles are formed now during this antral stage. So previous to this, they were arrested in the primary follicular stage in the meiotic cell prophase 1 of meiotic cell division, right? So now in every month we can say that these secondary follicles, they develop under the influence of the follicle stimulating hormone, or rather FSH, and luteinizing hormone, or LH. So these follicles are maturing in the presence of follicle stimulating hormone or luteinizing hormone in every month cycle, right? So this is the antral phase. Okay, now next comes the pre-ovulatory phase. So this stage is induced by the LH surge. So LH surge is happening and the meiosis one is completed here. Okay, two haploid cells of unequal sizes are formed within the follicle. So here secondary follicle is formed. Okay, due to LH surge, two unequal size haploid cells will be forming inside the follicle. So one is one of the daughter cells, they receive less cytoplasm and they form a very small polar body. Okay, and this cell does not participate in ovum formation. So ovum formation is from the secondary oocyte. A secondary oocyte is formed. The other daughter cell which is formed inside the follicle, secondary follicle, is the secondary oocyte. First one is the first small polar body is formed, which is not taking part in ovum formation, but another secondary oocyte is formed, and the two daughter cells, they undergo this meiosis 2. The polar body replicates to form the polar bodies, whereas the secondary oocytes arrests in the metaphase stage of meiosis 2. This again, it arrests in the metaphase stage of meiosis 2. So this is the pre-ovulatory phase. So then it is arrested in the meiosis 2. Now if fertilization is not happening, then this secondary oocyte, they all will then form the Graafian follicle and they will then rupture. Okay, so that we will see in the diagram in the next slide. So previous slides we have studied about the three phases: pre-ovulatory phase, antral phase, and where the we can say that pre-antral phase what was happening. So let us see, these are the primordial follicles, okay, or primary follicles. Okay, so after that, in the preantral phase, these preantral molecules are formed, and inside that we can say that zona pellucida is formed, and this antrum or fluid filled space is formed. Okay, now after that, this antral follicle, it is developing into antral follicle, and inside that we can see that matured ovum is there. Then this comes to the pre-ovulatory follicle. So this one, the pre-ovulatory follicles, where already the granulosa cells have developed. We are having the theca cells also, and inside that it is the corona radiata. So these all developments have happened from the preantral to the antral and to the pre-ovulatory follicle state. Okay, so at this phase they remain arrested, and then in the meiosis, we can say in the second phase of meiosis, they are arrested in metaphase 1 state, and then the ovulation happens. So this was in the pre-ovulatory states, then ovulation happens. Okay, this is the ovum formed along with the corona radiata. So if the sperm comes and fertilizes with the ovum, then fertilization and implantation will happen, or else what will happen? They will this will degenerate. Okay, so this degeneration of the endometrial wall will be happening, and that phase we will be studying in the next menstrual cycle phase, where we will be studying all the phases of menstrual cycle and how and why it happens in female from the stage of puberty to the stage of menopause. So it's a phase in the reproductive phase of female body where it is happening. Okay, so why and how it is happening we will be discussing in the next episode. So now we will move on to the whole summary of what we have studied in oogenesis. Let us go through that also. I think this diagram is clear to all of you. Please read and memorize this label diagram. This is very important for your exams, your NEET exams as well as your class exams. So please do go through it, and we will be coming in the summarization in the next slide. So just a quick recap of what we have studied till now. So it is according to your, as per your NCERT book. So let us see what is written in the book. So we will just follow what is written in the book, and we have already gone through the things which were to be discussed, like oogenesis and all. And now it is the process of formation of mature female gamete, which is called oogenesis. We already know that, which is markedly different from spermatogenesis. Now, oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cells or oogonia are formed within the fetal ovary. So when the ovary is in the fetal stage, then only oogonia are formed, and that too in a couple of million number. Now, they do not produce any more oogonia after birth or something. They remain the same, or they rather degenerate. These cells starts division and enter into prophase 1 of meiotic division and get temporarily arrested in this meiotic 1 phase at this stage. Okay, so this stage happens to be till the age of puberty. Okay, so this all primary oocytes, they remain in the prophase 1 of metaphase state, in the prophase 1 of the meiotic cell division till the puberty stage from the birth. So each primary oocyte then gets surrounded by a layer of granulosa cells and they form primary follicles. So they get surrounded by granulosa cells and it is called primary follicle. A large number of these follicles degenerate also. They were 2 million, we can say that couple of million gamete mother cells were there, oogonia, at the time of birth, but they degenerate during the phase from birth to puberty. During this phase they degenerate, and therefore at puberty only 60 to 80,000 primary follicles are left in each ovary. The primary follicle, surrounded by more layers of granulosa cells, new theca cells, they give rise to secondary follicles. Okay, so we have spoken in the previous slide that then comes the antral phase, that is the secondary follicle transformed into tertiary follicle, which is characterized by a fluid filled cavity. So we know already in the diagram in the previous slide, we know what is antrum. So the secondary follicle is maturing more into tertiary follicle, and that is having a fluid filled cavity called antrum. The theca layer is organized into an inner theca layer and an outer theca external layer. So inner theca internal layer and outer theca external layer. So theca has two parts. So it is important to draw your attention that is at this stage the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division. So now it is completing its first meiotic division at puberty, when the primary oocyte has been converted to the tertiary follicle. Now, in an equal division resulting in the formation of a large haploid secondary oocyte, we have spoken earlier, a haploid secondary oocyte and a tiny first polar body is being formed. And now we will say, now the secondary oocyte results bulk of nutrient-rich cytoplasm of the primary oocyte. Okay. So can you think of any advantage for this? Firstly, we can see that does the first polar body born out of the first meiotic division divide further or degenerate? So there are many questions we are having at present. We are not very certain about this. The tertiary follicle, which is formed, further changes into mature Graafian follicle. We can see in this diagram the primary follicle, we can see it here. It matures into the tertiary follicle, showing this space which is fluid filled space called antrum. Then again it is forming the Graafian follicle, and the secondary oocyte forms a new membrane called the zona pellucida. So it is forming a new membrane. This membrane is called the zona pellucida surrounding it. In the previous diagram, also in the previous slide, I have discussed about zona pellucida which is surrounding it. Then comes the Graafian follicle, which ruptures to release the secondary oocyte. So ovum or secondary oocyte is released from the ovary by the process of ovulation. So what is ovulation? The release of secondary oocyte or ovum from the ovary by the process of ovulation. So can you now identify the major differences between spermatogenesis and oogenesis? So this only we are going to discuss now. It's very important. So let us discuss. So what is the difference between spermatogenesis and oogenesis? So the first prime difference is spermatogenesis is the production of sperms from the male germ cells and spermatogonia in males, while oogenesis is the production of eggs from the female germ cells or oogonia in females. So this is the basic difference. We all know that. Spermatogenesis happens in testes, oogenesis occurs in ovaries. Second one. Okay, third one, spermatogenesis is a continuous process while oogenesis is a discontinuous process. It was arrested from birth till puberty. It was in the prophase 1 of meiotic cell division phase. So it was arrested. The early stages of oogenesis occur before birth and the final stages occur after puberty. So it is a discontinuous process. Is it clear to everyone? Please remember these points specifically so that it will be very clear for you all. Now spermatogenesis produces 4 haploid sperms from 1 haploid spermatogonium, while oogenesis produces 1 haploid egg and 3 polar bodies from 1 diploid oogonium. Okay, so it is producing 1 haploid egg and 3 polar bodies from 1 diploid oogonium. Is it clear? So from 1 diploid oogonium, 3 polar bodies and 1 haploid egg is produced. But during spermatogenesis, 1 diploid spermatogonium is producing 1 haploid spermatid. Okay, so like that the process is happening. So is it clear? There are major differences between spermatogenesis and oogenesis. So now we will move on with the diagram of spermatogenesis and oogenesis. So let us see now what is happening in both the processes, spermatogonium and oogonium. These are two we have taken. So this is spermatogenesis. This is the process of spermatogenesis and this is the process of oogenesis. So by this diagram we will make it clear what is happening in these two processes. So this is spermatogonium, this is oogonium. In the first growth phase we see that in growth and maturation phase both are producing primary oocyte and primary spermatocyte. Okay, primary spermatocyte and primary oocyte is formed. Now in meiosis 1 when it is happening, they are producing secondary spermatocytes. 2 secondary spermatocytes are produced and they are haploid because it's a reductional division. Okay. Now here also one secondary oocyte is formed and a first polar body is formed. But this phase it remains arrested in this phase till the puberty is reached. But here it starts only in the puberty. So it is a continuous process. This process will be a continuous process, whereas this process is not a continuous process. It is arrested in this prophase 1 of meiosis and this will happen after puberty. So this phase happens after puberty. Okay. So then again in the meiosis 2, this secondary spermatocytes, they produce spermatids. So 4 spermatids are formed, but here 1 ootid and 1 second polar body is formed. First polar body was formed here. Again from the secondary oocyte in the meiosis 2, there is 1 ootid and 1 second polar body is formed. Now again during this differentiation phase, whereas in this spermatids they all mature to spermatozoa or sperms, mature sperms or spermatozoa. 4 are formed, 1 from each spermatid. But in this oogenesis process, from this ootid, 1 ovum is formed only. Okay, so this is the clear difference between spermatogenesis and oogenesis process. I have also discussed that in the previous slide, and this diagram is very important. Just go through this diagram, learn this, memorize this, then it will be very easy for you to answer any questions either in your NEET or in your board exams, right? So stay tuned, stay updated because many more important lessons are yet to come as per your CBSE curriculum. And please don't skip my lessons. Please do go through the lessons in full because many important things are there being taught in the middle as well as in the end. Stay tuned, stay updated and share my videos with your friends as much as you can. Thank you and please go through this lesson again before I come back with the next part of menstrual cycle. Thank you.